Hello everyone, welcome back to Grand Archive classes. In my previous lecture, I have discussed about the cloning vector. Now in this lecture, let's see what is competent host, meaning for transformation of recombinant DNA. Before starting this, there is one question in your NCERT books chapter. See, DNA is hydrophilic molecule. It cannot pass through cell membrane. Why? Because DNA, it is basically from nucleus, not from cytoplasm. Okay. C, it basically prevent from the various type of the enzymes and protect it to reaching to the membrane. That is the main reason why this is known. It did not reach to the membrane. Now see, there are certain steps such as treating with chemical treatment. Using a divalent calcium ion increases the efficiency and to reach to the host cell. Okay, it is now clear. Now see, second point, incubating both on ice or followed by placing them briefly at 42 degrees Celsius, then putting back on ice. It helps to reach RDNA to the host. See, now macro injection. It is basically that RDNA is transformed into the host by using a glass micropipette. It is now clear. Now see, biolistic or gene gun. It is basically used to inject RDNA to the host in plant or animal. Now see, fifth point, disarm pathogen vector. It is basically inject to the cell to transform RDNA to the host cell. Now see, process of recombinant DNA technology. It is basically that DNA molecule joined together form a two different species and it is now inserted to the host cell to produce a new genetic combination. It is basically used in the field of the agriculture. Now see, recombinant DNA technology is basically involves several steps. First one is that isolation of DNA. Okay. Second step is that fragmentation of DNA by restriction into nucleus. This restriction into nucleus I have explained you in my previous lecture. Now, third step is that isolation of adversarial DNA fragment. Okay, or a desired DNA fragment. Now, fourth point is that ligation of the DNA fragment into vector. What is a ligation? That I have also explained you. Now, fifth point is that transforming the rDNA into the host. This I have also discussed in my previous lecture. Now, sixth point is that culturing the host cell in the medium at the large scale. Seventh and the last, extraction of the desired product, which may be useful in the any field of the biotechnology. Okay, it is now clear. Now see, isolation of genetic material from macromolecules by using enzymes such as lysozyme found in bacteria, cellulose in plant cells, chitinase in fungi. Okay, let's see what is an lysozyme. It is a naturally occurring enzyme found in bodily secretion of such as tears and saliva. It is now clear. Cellulose is a anisexual enzyme produced chiefly by fungi, bacteria, protozoa that catalyze cellulolysis, the decomposition of cellulose and some related polysaccharide. It is now clear. Now see, chitinase, all the hydrolytic enzymes that break down the glycosidic bonds in the chitin. Okay, it is now clear what is the meaning of these three enzymes. Okay. Now see, cutting of DNA at a specific location. It is basically by two methods. First one is restriction enzyme. Second one is agarose gel electrophoresis. Do you have remember that I have explained about you this in my previous lecture? See, basically DNA has been cut from certain specific location. It basically get inserted into the new host cell. Then it start replicating and developing and making a number of copies in that. Now see, amplification of gene inserting using a PCR. What is the meaning of the PCR? PCR is basically defined as polymerase chain reaction. It is now clear. See, Chain reaction, by the name it is clear, is a method of widely used to rapidly make millions or billions of copy of specific DNA. See, chain reaction means making a chain of that thing, making a multiple copies into millions, billions and tens of trillions also. See, it is now clear. Now see about the PCR. This double stranded DNA has been denatured by heating. By heating, it just simply break down in each single strand. See, this is a primer. Now with the first it has been denatured. Now after the annealing, then DNA polymerase plus deoxynucleotide come together and join it and call extension of it. Now see, this by replicating form 30 cycle, and after the amplified they form billions of tons of copies. It is very clear what is the meaning of polymerase chain reaction to creating a billion or trillion of copies by simply in three steps: by denaturing, number one, second one is annealing, number two, and third one is extension, number third. Okay, it is now clear. Now see, insertion of our DNA into a host cell or organisms. It is done by selectable marker. What is a selectable marker? See, is a gene introduced into a cell, especially a bacteria to cell in culture, that confirms a trait suitable for artificial selection. Simply, it has meaning that if the genes is introduced into the cell, okay, by implementing in the culture, that we see the response what does happens is suitable for the artificial selection or not. Okay, it is not clear. See, next point, obtaining the foreign DNA gene products. See, foreign gene products that has been obtained by certain. See, the foreign DNA multiply in plant or animal cell to produce desirable protein. Expression of the foreign gene in host cell involve optimizing condition to obtain recombinant protein. It is simple thing that foreign DNA multiply in plant or animal cells. Basically, they produce desirable proteins by their multiplication. Okay, now it is clear to all. Now see about the bioreactor. By the name it is clear. Bio plus reactor. See, there are two types. First one is simple stirred tank bioreactor. Second one is that sparge stirred tank bioreactor. See, simple stirred tank bioreactor. It is composed of a reactor and the mixer such as stirred a turbine, wing or a propeller. It is useful for substrate solution of high viscosity and for immovable enzymes which relative low activity, which so basically low activity. Now see second point is past stirred tank reactor aerobic cultivation process. The popular of the sparger is to supply adequate oxygen to the growing cell because it is very important for the growing cell need of the oxygen also metabolize in their growth also. Now see about the downstream process. By the name it is clear. See, refers to the recovery or purification of biosynthetic product, particularly pharmaceutical form or natural resources. It is now clear. Now see, now see, this time I have completed my the lecture of the first chapter, principle and process of biotechnology. I will give you question in my next lecture. Please like, subscribe or share this video. Thanks.